Welcome everyone. Today I'll be explaining question number 43 of the 2022 GATE Biotechnology paper. And the question is that from the given options, you have to choose the enzymes involved in ubiquitination of cell cycle protein. There are four options given to you. Option A is E1 and E2 only. Option B is E1 and E3 only. Option C is E1 and E4 only. And option D is E1, E2 and E3. So before we move on, let's see why there is a need for ubiquitination of cell cycle protein. So as you know that the progression through the cell cycle is determined the phosphorylation of something called CDK substrate. Okay? And to ensure the progression, cell cycle progression, there is an oscillating activity of CDK, which is controlled by a few mechanisms like transcription and phosphorylation, as well as the periodic degradation, that is because of ubiquitination of proteins called cyclin and CKI. Okay? The periodic degradation of these cyclins and CKIs is due to the ubiquitination of them, which targets them for degradation by proteasomes. Right? So now that we have understood why ubiquitination is required, let's move on to understand the process involved. So there are three enzymes involved in the process of ubiquitination in cell cycle proteins. It is E1, E2, and E3. Okay? So E1 is a ubiquitin activating enzyme, E2 is a ubiquitin conjugating enzyme, and E3 is a ubiquitin ligase. So the first step is activated, that is mediated by E1. So what happens is there is a E1 mediated catalysis of the acyl adenylation of the C terminus of the ubiquitin protein. Okay, and this is followed by the transfer of this ubiquitin to the active site of E1, which is cysteine. Okay, and this is done with the help of ATP. So there is energy involved. The second step is that E2 ubiquitin conjugating enzyme, which is this enzyme, will catalyze the transfer of ubiquitin from E1 to the active site, that is again a cysteine of E2, to form a E2 ubiquitin complex. And finally, the E3 ubiquitin ligase will bring the substrate, okay, and the ubiquitin loaded E2 together in this case, okay, and catalyze the transfer of ubiquitin from E2 to substrate to form a ubiquitin substrate complex. Okay? And this will further down the road be degraded by proteases. So the correct answer in this case would be option number D, which is E1, E2, and E3 enzymes. So these are the enzymes which are involved in the ubiquitination of cell cycle proteins. So that is all for this question. I hope that was easy for you to understand. Thank you so much for listening.